Okay, folks, just one more set of tests to talk about here, and these are not auger plates, not slants, which are basically auger inside of tubes, but we're back to broth, okay? And we only have a couple different broth tests to look at here. There are a couple different names used for these particular assays. One, we might just say they're fermentation tubes, but that wouldn't be very accurate. We could be a little more accurate and say carbohydrate fermentation tubes, which would indicate that there is some kind of a sugar inside of the tube dissolved in that broth as an energy source. But to be even more accurate, we could say that there are phenol red lactose fermentation tubes. Okay, so we have the lactose in there. That is the specific carbohydrate. Or we could say phenol red sucrose broth or fermentation tube. And that would be, to use the terms lactose or sucrose would tell you exactly what carbohydrate is dissolved in that medium as a resource. Okay? Now, we always want to know what are the key ingredients in every medium, whether it's an auger, a slant, or a broth tube. And in this case, what we have is the carbohydrate, which is either lactose or sucrose. In our discussions, those are what we will limit ourselves to. But it could be anything under the sun, really. We could use any sugar in here. Glucose, we could really maltose, go on and on, okay? But for us, we're going to just stick to these two, lactose and sucrose as the sugars, and there's a phenol red pH indicator. Now over on the left, we see an uninoculated broth tube, and it is red, just like the mannitol salt auger was, right? And in the mannitol salt auger, it started red, it was neutral. Same here. This is a neutral plate to get started, right? And we're looking for a color change to see if something was fermented. Now we also see something different about these tubes. This is not just one tube here over on the left. We see this other tube that's kind of oriented upside down. See if I can draw this without messing it up here. So we have this little upside down tube on the inside of the bigger tube. And it may not be real obvious here, but the red medium that's in here that is either containing lactose or sucrose, this red medium is all the way up in this tube, this little tube that's upside down, okay? This is called a Durham tube. All right, so every one of these tubes, when they start out, have a big tube with a broth and then an upside down Durham tube inside there that's filled up with that broth as well. Okay, now the tubes would be inoculated, right? So we go and we get a sample, put the inoculum into each of these tubes, cap the tube off. Usually these tubes wouldn't be capped tightly because we want to let air in there, right? So we'd loosen the tube, stash it in the incubator, come back 24 hours later, see what happened, okay? See if anything happened to these tubes, if anything grew or not. All right, so here we have A, B, and C, different results that you might expect to find coming back the next day. Now, B doesn't look to have changed very much. Number one, it's still red. There's nothing different about the little upside down tube here. It still has red medium in it. And as long as there's no color change, we can say that the organism that you inoculated this tube with is not capable of fermenting that sugar. Now, if this, let's say this is a lactose tube, it has lactose, that's the only thing. Then we say it's a I'll write it out here, okay? We'd say that this is a non-lactose fermenter, if lactose is the sugar we're using, okay? Now, we see some very different results on the A and the B example, right? We do see some yellow in both cases, and that tells us something. That tells us that the phenol red, okay, is no longer red, right? So now we're getting this yellow color from the phenol red, and that means that acids were produced. There was fermentation, in other words, okay? So we can say in both of these cases there is lactose fermentation. But that's not all I want to say about these two tubes. We want to differentiate. This is the whole point, right? We're doing all of these plates, all of these slants, all of these broths to differentiate, to tell these organisms apart, okay? So I see here with this particular Durham tube, there's a little bit of gas in there. Versus over here, there's no gas at all in the Durham tube in selection A. So what I would say to be more accurate and more complete about lactose fermentation in tubes A and C is I would write A, it's a lactose fermenter for choice A, and I'd write A for acids that were produced there, okay? Over in tube C, I don't just see a color change that would indicate an acid being produced, acidic byproducts as a result of fermentation, but I'd say AG, that there was acids and there was a gas produced that got trapped in that little upside down tube. And that's exactly the point of that little upside down tube. That little Durham tube is there to catch gas so that we can tell the difference between a lactose fermenter that doesn't give off a gas, like A, and a lactose fermenter that does produce gas as a byproduct in C. All right? So if there's yellow medium, you'd say either lactose fermenter or sucrose fermenter, indicating that it's an acid that's being produced. If it's yellow with the gas in the Durham tube, we'd say lactose fermenter AG or sucrose fermenter AG. Okay? Let me show you just one more example of fermentation tubes. So here we have some tubes. I'm not sure what sugar they used, quite honestly, in these tubes. But I can tell that there's some yellow going on with E. coli, and there's some yellow going on with this particular organism that I'm not as familiar with here. Definitely we have some fermentation going on in these two in the middle. Now the uninoculated tube doesn't look particularly red. I'll be honest, I would agree with you if you said, well, that's not red. You'd be right. But it's clearly not yellow, and that's what we need to look for. Okay, so if the inoculated tube looks red, orange, well, it's all the same, right? Nothing happened. There was no fermentation there. And for that reason, looking over here at this particular tube, this is Pseudomonas. The P is for Pseudomonas, okay? Pseudomonas causes problems with catheter infections. It'll grow on your contacts if you let them go too far. Pseudomonas can be a problem for cystic fibrosis patients too. They can get into their lungs and really cause problems. Now it looks to me that Pseudomonas is just showing us this kind of orange color, just like the original tube. So this would be a negative for fermentation here in this fourth tube versus a positive for fermentation, these two middle ones. And what's more, we could say that E. coli ferments its sugar really well and makes acids and gases, versus this third one, which only makes acids in its fermentation process. Okay? All right, so there are a lot of media here, Guys, I'm going to give you some hints, and I'm going to start by just suggesting to you guys that you make a chart for all of these media and fermentation tubes and things like that. A chart where you'd have a column for the test, a column for what are the key ingredients contained within the media, a column for the type of medium that it is it complex, selective, differential, and then two more columns. One would be what are the different observations that you might see as a result of running each test, and what do each of those observations tell you? What is the interpretation of that change? Okay. So this is one way to prepare, and I would fully encourage you to use that chart in this next lab quiz. Okay. I'm usually thinking that you're probably not going to have time to look up everything during the quiz. In this case, I fully encourage you to put a chart together with name, contents, type of medium, the observations you might see, and the interpretations of those observations. All right. Again, let me know if you have trouble. If you want me to go over stuff with you, I can easily get together with you in Blackboard or up on Google Meet or something, or Zoom. Happy to do so. Okay.